CSIR Evolution Chapter 5. Chapter 5. The History of Life. The history of life on Earth traces the processes by which living and fossil organisms evolved, from the earliest emergence of life to the present. Earth formed about 4.54 billion years ago, and evidence suggests that life emerged prior to 3.7 billion years ago, and by about 1.5 billion, eukaryotic organisms have evolved. The similarities among all known present-day species indicate that they have diverged through the process of evolution from a common ancestor. 5.1 Geological Time Scale. Fossils are a major source of information about changes on Earth during the remote past. Pre-Darwinian geologists divided geological history into units based on their distinct fossil assemblages of animals, Table 4 and 5. These divisions are marked either by mass extinctions or by dramatic increases in the diversity of major groups of organisms, called evolutionary radiations. The earliest evidence of life comes from biogenic carbon signatures and stromatolite fossils discovered in 3.7 billion-year-old metasedimentary rocks from western Greenland. In 2015, possible remains of biotic life were found in 4.1 billion-year-old rocks in Western Australia. In 2017, putative evidence of possibly the oldest forms of life on Earth was reported in the form of fossilized microorganisms discovered in hydrothermal vent precipitates in the Nuvvuagittic belt of Quebec, Canada, that may have lived as early as 4.28 billion years ago, not long after the oceans formed 4.4 billion years ago, and not long after the formation of the Earth 4.54 billion years ago. The fossil record of organisms that lived prior to 550 Maya is fragmentary, but the available evidence suggests that the major divisions in many animal lineages predate the end of the Precambrian by more than 100 million years. The fossil record shows that the total number of species and individuals increased dramatically in late Precambrian times. An organism is most likely to become a fossil if its dead body is deposited in an environment that lacks oxygen. However, most organisms live in aerobic environments. They decompose completely when they die. Thus, many fossil assemblages are collections of organisms that were transported by wind or water to sites that lacked oxygen. Occasionally, organisms are preserved where they lived. In such cases, especially if the environment in question was a cool, anaerobic swamp, where conditions for preservation were excellent. About 300,000 species of fossil organisms have been described, and the number is growing steadily. However, this number is only a tiny fraction of the species that have ever lived. We do not know how many species lived in the past, but we have ways of making reasonable estimates. Approximately 1 trillion species currently live on Earth, of which only 1.75 to 1.8 million have been named, and 1.8 million documented in a central database. These currently living species represent less than 1% of all species that have ever lived on Earth. Because life has existed on Earth for about 3.8 billion years, and because species last, on average, less than 10 million years, Earth's biota must have turned over many times during geological history. Thus, the total number of species that lived over evolutionary time must vastly exceed the number living today. The number of known fossils, although it is a small fraction of the total number of extinct species, is especially large for marine animals that had hard skeletons. Among the 9 major animal groups with hard-shelled members, approximately 200,000 species have been described. From fossils, roughly twice the number of living marine species in these same groups. Paleontologists, scientists who study fossils, lean heavily on these groups in their interpretations of the evolution of life. Insects and spiders are also relatively well represented in the fossil record. The fossil record may be incomplete, but it is good enough to demonstrate clearly that organisms of particular types are found in rocks of specific ages, and that new organisms appear, sequentially, in younger rocks. Fossil can only sets a minimum age for a lineage. Presence of fossil confirms the organism at least for that period, but absence of fossil does not. Means that organism were not present. It may be there, but may not be fossilized, or fossils may have not been recovered. The fossil record also tells us that extinction is the eventual fate of all species. By combining information about physical changes during Earth's history with evidence from the fossil record, scientists have composed portraits of what Earth and its inhabitants may have looked like at different times. The geological time scale is a system of chronological dating that classifies geological strata, stratigraphy, in time. It is used by geologists, paleontologists, and other Earth scientists to describe the timing and relationships of events in geologic history. The primary and largest cataloged divisions of time are periods called eons. The first eon was the Hadean, starting with the formation of the Earth and lasting over 600 million years until the Archean, which is when the Earth had cooled enough for continents and the earliest known life to emerge. After about 2.5 billion years, oxygen generated by photosynthesizing single-celled organisms began to appear in the atmosphere, marking the beginning of the Proterozoic, Table 4. Finally, the Phanerozoic eon encompasses 541 million years of diverse abundance of multicellular life, starting with the appearance of hard animal shells in the fossil record and continuing to the present, Table 5. Eons are divided into eras, which are in turn divided into periods, epochs, and ages. The first three eons, i.e. every eon but the Phanerozoic, can be referred to collectively as the Precambrian supereon. This is in reference to the significance of the Cambrian explosion, a massive diversification of multicellular life forms that took place in the Cambrian period at the start of the Phanerozoic. 1.2 Oxygen Revolution. The first living beings on Earth were anaerobic because there was no oxygen in air. Most atmospheric oxygen gas, O2, is of biological origin, produced during the water-splitting step of photosynthesis. When oxygenic photosynthesis first evolved, the free O2 it produced probably dissolved in the surrounding water until it reached a high enough concentration to react with dissolved iron. This would have caused the iron to precipitate as iron oxide, which accumulated as sediments. These sediments were compressed into banded iron formations, red layers of rock containing iron oxide that are a source of iron ore today. Once all of the dissolved iron had precipitated, additional O2 dissolved in the water until the seas and lakes became saturated with O2. After this occurred, the O2 finally began to gas out of the water and enter the atmosphere. This change left its mark in the rusting of iron-rich terrestrial rocks, a process that began about 2.7 billion years ago. This chronology implies that bacteria similar to today's cyanobacteria, oxygen-releasing, photosynthetic bacteria, originated well before 2.7 billion years ago. The amount of atmospheric Oz increased gradually from about 2.7 to 2.3 billion years ago, but then shot up relatively rapidly to between 1% and 10% of its present level, Figure 32. Figure 32. The rise of atmospheric oxygen due to photosynthetic activity of cyanobacteria. This oxygen revolution had an enormous impact on life. In certain of its chemical forms, oxygen attacks chemical bonds and can inhibit enzymes and damage cells. As a result, the rising concentration of atmospheric O2 probably doomed many prokaryotic groups. Some species survived in habitats that remained anaerobic, where we find their descendants living today. Among other survivors, diverse adaptations to the changing atmosphere evolved, including cellular respiration, which uses O2 in the process of harvesting the energy stored in organic molecules. As mentioned previously, the early, gradual rise in atmospheric O2 levels was probably brought about by ancient cyanobacteria. A few 100 million years later, the rise in Oz accelerated. One hypothesis is that this rise followed the evolution of eukaryotic photosynthetic cells containing chloroplasts. 1.3 The origin of eukaryotes. Complex eukaryotic cells may have evolved from simpler prokaryotic cells because complexity of organization increases the ability to attain large size. The oldest widely accepted fossils of eukaryotic organisms are about 2.1 billion years old. Recall that eukaryotic cells have more complex organization than prokaryotic cells. Eukaryotic cells have a nuclear envelope, mitochondria, endoplasmic reticulum, and other internal structures that prokaryotes lack. Also, unlike /D prokaryotic cells, eukaryotic cells have a cytoskeleton, a feature that enables eukaryotic cells to change their shape and thereby surround and engulf other cells. How did these eukaryotic features evolve from prokaryotic cells? A range of evidence supports the endosymbiont theory, which posits that mitochondria and plastids, a general term for chloroplasts and related organelles, were formerly small prokaryotes that began living within larger cells. The term endosymbiont refers to a cell that lives within another cell, called the host cell. The prokaryotic ancestors of mitochondria and plastids probably gained entry to the host cell as undigested prey or internal parasites. Though such a process may seem unlikely, scientists have directly observed cases in which endosymbionts that began as prey or parasites came to have a mutually beneficial relationship with the host in as little as 5 years. By whatever means the relationships began, we can hypothesize how the symbiosis could have become mutually beneficial. A host that is a heterotroph, an organism that eats other organisms or substances derived from them, could use nutrients released from photosynthetic endosymbionts. In a world that was becoming increasingly aerobic, a host that was itself an anaerobe would have benefited from endosymbionts that turned the oxygen to advantage. Over time, the host and endosymbionts would have become a single organism, its parts inseparable, although all eukaryotes have mitochondria or remnants of these organelles, they do not all have plastids. Thus, the hypothesis of serial endosymbiosis supposes that mitochondria evolved before plastids through a sequence of endosymbiotic events. Figure 33. A great deal of evidence supports the endosymbiotic origin of mitochondria and plastids. The inner membranes of both organelles have enzymes and transport systems that are homologous to those found in the plasma membranes of living prokaryotes. Mitochondria and plastids replicate by a splitting process that is similar to that of certain prokaryotes. In addition, each of these organelles contains a single, circular DNA molecule that, like the chromosomes of bacteria, is not associated with histones or large amounts of other proteins. As might be expected of organelles descended from free-living organisms, mitochondria and plastids also have the cellular machinery, including ribosomes, needed to transcribe and translate their DNA into proteins. Finally, in terms of size, RNA sequences, and sensitivity to certain antibiotics, the ribosomes of mitochondria and plastids are more similar to prokaryotic ribosomes than they are to the cytoplasmic ribosomes of eukaryotic cells. Figure 33. Evolution of present eukaryotic cells by mechanism of serial endosymbiosis. 4. The origin of multicellularity. The appearance of structurally complex eukaryotic cells sparked the evolution of greater morphological diversity than was possible for the simpler prokaryotic cells. After the first eukaryotes appeared, a great range of unicellular forms evolved, giving rise to the diversity of single-celled eukaryotes that continue to flourish today. Another wave of diversification also occurred. Some single-celled eukaryotes gave rise to multicellular forms, whose descendants include a variety of algae, plants, fungi, and animals. Based on comparisons of DNA sequences, researchers have suggested that the common ancestor of multicellular eukaryotes lived 1.5 billion years ago. This result is in rough agreement with the fossil record. The oldest known fossils of multicellular eukaryotes are of relatively small algae that lived about 1.2 billion years ago. Larger and more diverse multicellular eukaryotes do not appear in the fossil record until about 575 million years ago. These fossils, referred to as the Ediacaran blota, were of soft-bodied organisms some over 1 m long that lived from 575 to 535 million years ago. Why were multicellular eukaryotes limited in size and diversity until the late Proterozoic? One hypothesis stems from geologic evidence, indicating that a series of severe ice ages occurred from 750 to 580 million years ago. At various times during this period, glaciers covered all of the planet's landmasses, and the seas were largely iced over. The snowball Earth hypothesis suggests that most life would have been confined to areas near deep-sea vents and hot springs or to equatorial regions of the ocean that lacked ice cover. The fossil record of the first major diversification of multicellular eukaryotes, beginning about 575 million years ago, corresponds roughly to the time when snowball Earth thawed. As that diversification came to a close about 40 million years later, the stage was set for another, even more spectacular burst of evolutionary change. For much of its history, life was confined to the oceans, and all organisms were small. In the Precambrian era, shallow seas teemed with life. Protists and small multicellular animals fed on floating algae. Small floating organisms, known collectively as plankton, were eaten by small animals that filtered them from the water. Other animals ingested sediments on the bottom of the seas and digested the remains of organisms within them. By the late Precambrian, about 650 Maya, many kinds of soft-bodied invertebrates had evolved. Some of them were very different from any animals living today. They may be members of animal lineages that have no living descendants. 5.5 The Paleozoic Era. Geologists divide the Paleozoic era into 6 periods: the Cambrian, Ordovician, Silurian, Devonian, Carboniferous, and Permian, Table 5. Each period is characterized by the diversification of specific modern fauna such as bivalves, gastropods, bryozoans, malacostrican crustaceans. As extinctions mark the ends of 3 periods: the Ordovician, Devonian, and Permian. 5.5.1 The Cambrian period. By the early Cambrian period, 543 to 490 Maya, at the beginning of the Paleozoic era, the O2 concentration in the atmosphere approached its current level, and the continental plates came together to form several land masses. The largest of the land masses was called Gondwana, figure 34. All of the major groups of animals that have species living today appeared during the Cambrian. This rapid diversification of life is referred to as the Cambrian explosion. The most extensive fossil evidence from the Cambrian comes from the unusually well-preserved animal fossils recently discovered in northeastern China. Figure 34. Position of continents during Mid-Cambrian time. Arthropods, crabs, shrimps, and their relatives are the most diverse group in this Chinese fauna. Some of them were large carnivores. Trilobites, an arthropod group that was abundant and diverse during the Cambrian, suffered a major extinction at the end of the Cambrian. but they recovered and continued to be abundant during subsequent periods. Figure 35. Appearance of selected animal groups. Bars indicate earliest appearance in the fossil records. 5.5.2 The Ordovician, 490 to 443 MYA. During the Ordovician period, the continents, which were located primarily in the southern hemisphere, were still devoid of multicellular plants. Evolutionary radiation of marine organisms was spectacular during the early Ordovician, especially among animals that filter small prey from the water, such as brachiopods and mollusks, and diversification of echinoderms was observed. The first vertebrates appeared in the form of jawless fish, which greatly diversified in the Silurian and Devonian periods. All animals lived on the seafloor or burrowed in its sediments. At the end of the Ordovician, as massive glaciers formed over Gondwana, sea levels dropped about 50 meters and ocean temperatures dropped. About 75% of the animal species became extinct, probably because of these major environmental changes. First, primitive plants, bryophytes, without vascular tissue, appeared and colonized lands. Fungi also colonized land. 9.5.3 The Silurian, 443 to 417 Maya. During the Silurian period, the northern continents coalesced, but the general positions of the continents did not change much. Marine life rebounded from the mass extinction at the end of the Ordovician. Animals able to swim and feed above the ocean bottom appeared for the first time, but no new phyla of marine organisms evolved. The tropical sea was uninterrupted by land barriers, and most marine genera were widely distributed. On land, the first known trichiophytes, plants with true vascular tissue, appeared late in the Silurian period, about 420 Maya. These plants, in the genus Cooksonia, were less than 50 centimeters tall and lacked roots and leaves and are true land plants. The first terrestrial arthropods, insects, scorpions and millipedes appeared at about the same time. 5.6.3 The Devonian, 417 to 354 Maya. Rates of evolutionary change accelerated in many groups of organisms during the Devonian period. Both northern and southern land masses slowly moved northward. Figure 36. There were great evolutionary radiations of corals and shelled squid-like cephalopods. Trilobites were abundant. Fishes diversified as jawed forms replaced jawless ones, and heavy armor gave way to the less rigid outer coverings of modern fishes. All current major groups of fishes were abundantly present by the end of the period. Terrestrial communities also changed dramatically during the Devonian. Club mosses, horsetails, and tree ferns became common toward the end of the Devonian. Some attained the size of trees. Their deep roots accelerated the weathering of rocks, resulting in the development of the first forest soils. Distinct forests evolved on the two major land masses toward the end of the period, and the ancestors of gymnosperms, the first plants to produce seeds, appeared in the fossil record. The first known fossils of centipedes, spiders, mites, and insects date to this period. First-time fish-like amphibians, earliest amphibians, began to occupy the land. An extinction of about 75% of all marine species marked the end of the Devonian. Paleontologists are uncertain about the cause of this mass extinction, but two large meteorites that collided with Earth at that time, one in present-day Nevada and the other in western Australia, may have been responsible. Figure 36. During the Devonian period, the northern and southern continents were approaching one another. 5.6.4 The Carboniferous, 354 to 290 Maya. Large glaciers 6.4 formed over high-latitude Gondwana during the Carboniferous period, but extensive swamp forests grew on the tropical Continents. These forests were not made up of the kinds of trees we know today, but were dominated by giant tree ferns and conifers. Pteridophytes/gymnosperms. Fossilized remains of those trees formed the coal we now mine for energy. The diversity of terrestrial animals increased greatly. Snails, scorpions, centipedes, and insects were abundant and diverse. Insects evolved wings, becoming the first animals to fly. Flight gave them access to tall plants. Plant fossils from this period show evidence of chewing by insects. True amphibians appeared and became larger and better adapted to terrestrial existence. From one amphibian stock, the first reptiles evolved late in the period. In the seas, crinants, sea lilies, and feather stars reached their greatest diversity, forming meadows on the seafloor. 5.6.5 The Permian, 290 to 248 Maya. During the Permian period, the continents coalesced into a supercontinent called Pangaea. Massive volcanic eruptions resulted in outpourings of lava that covered large areas of Earth. Figure 37. The ash they produced blocked sunlight and cooled the climate, resulting in the largest glaciers in Earth's history. Permian deposits contain representatives of most modern groups of insects. By the end of the period, adaptive radiation of reptiles greatly outnumbered amphibians. At the same time, there was decline in amphibian fauna. Late in the period, the lineage leading to mammals diverged from one reptilian lineage. In fresh waters, the Permian period was a time of extensive radiation of ray-fin fishes. Figure 37. Supercontinent, Pangaea, formed in the Permian period. Toward the end of the Permian period, a large meteorite crashed into northwestern Australia. In addition, a massive outpouring of lava flowed into the oceans, drastically reducing the oxygen content of deep ocean waters. Oceanic turnover then carried these oxygen-depleted waters toward the surface, where they released toxic concentrations of carbon dioxide and hydrogen sulfide into the surface waters and the atmosphere. These gases poisoned most of the species that had survived the impact. All in all, about 96% of all species on Earth became extinct at that time. 5.6 The Mesozoic Era. The few organisms that survived the Permian mass extinction found themselves in a relatively empty world at the start of the Mesozoic Era, 248 Maya. As Pangaea slowly separated into individual continents, the climate warmed, the glaciers melted, and the oceans rose and reflooded the continental shelves, forming huge, shallow inland seas. Life again proliferated and diversified, but different lineages came to dominate Earth. There was dominance of reptiles among animals. The pteridophytes trees that had dominated the Permian forests were replaced by plants with seeds, gymnosperms. At the end of Mesozoic, flowering plants, angiosperms, mammals, and birds were present. During the Mesozoic, Earth's biota, which until that time had been relatively homogeneous, became increasingly provincialized, that is, distinctive terrestrial floras and faunas evolved on each continent. The biotas of the shallow waters bordering the continents also diverged from one another. The provincialization that began during the Mesozoic continues to influence the geography of life today. By the end of the era, the world and its biota appeared quite modern. The Mesozoic era is divided into three periods: the Triassic, Jurassic, and Cretaceous. 5.6.1 The Triassic, 248 to 206 Maya. Pangaea began to break apart during the Triassic period. Many invertebrate lineages became more diverse, and many burrowing forms evolved from groups living on the surfaces of bottom sediments. On land, conifers and seed ferns became the dominant trees. The first frog, turtles, and mammals appeared. A great radiation of reptiles began, which eventually gave rise to crocodilians and dinosaurs. The end of the Triassic was marked by a mass extinction that eliminated about 65% of the species on Earth. 5.6.2 The Jurassic, 206 to 144 Maya. During the Jurassic period, two large continents formed: Laurasia in the north and Gondwana in the south. Diversification of many lineages proceeded. Ray-finned fishes began the great radiation that culminated in their dominance of the oceans. Salamanders and lizards first appeared. Flying reptiles, pterosaurs, and earliest birds evolved, and dinosaur lineages evolved into bipedal predators and large quadrupedal herbivores. Several groups of mammals appeared during this time. Plant evolution continued with the likely emergence of the ancestors of flowering plant, angiosperm, lineage, prevalent on Earth today. 5.6.3 The Cretaceous, 144 to 65 Maya. By the early Cretaceous period, Laurasia was completely separate from Gondwana, which was beginning to break apart. A continuous sea encircled the tropics, figure 38. Sea levels were high, and Earth was warm and humid. Life proliferated, both on land and in the oceans. In this period, flowering plants, angiosperms, appeared. Marine invertebrates increased in variety and number of species. On land, dinosaurs continued to diversify. The first snakes appeared during the Cretaceous, though their lineages did not radiate until much later. Cretaceous is known for angiosperm diversification. Early in the Cretaceous, flowering plants, angiosperms, began the radiation that led to their current dominance on land. Darwin's abominable mystery is about the origin of all angiosperms, suddenly and in great diversity in the Cretaceous, with no obvious ancestral lineage. Fossils of the earliest known angiosperm, Archaefructaceae, dated at 124 Maya, were discovered in Liaoning province in northeastern China. By the end of the period, many groups of mammals had evolved, but these mammals were generally small. Another meteorite-caused mass extinction took place at the end of the Cretaceous period. On land, all vertebrates larger than about 25 kilograms in body weight, including all of the dinosaurs, apparently became extinct. Many species of insects died out, perhaps because the growth of their food plants was greatly reduced following the impact. In the seas, many planktonic organisms and bottom-dwelling invertebrates became extinct. 5.7 The Cenozoic Era. By the early Cenozoic era, 65 Maya, the positions of the continents resembled those of today, but Australia was still attached to Antarctica, and the Atlantic Ocean was much narrower. The Cenozoic era was characterized by an extensive radiation and diversification of mammals. But other groups were also undergoing important changes. Flowering plants diversified extensively and came to dominate world forests, except in cool regions. The Cenozoic era is divided into two periods, the Tertiary and the Quaternary. Figure 38. During Cretaceous, Pangaea split into two major land masses. 5.7.1 The Tertiary, 65 to 1.8 Maya. During the Tertiary period, Australia began its northward drift. By 20 Maya, it had nearly reached its current position. The early Tertiary was a hot/humid time, during which vegetation belts shifted latitudinally. The tropics were probably too hot for rainforests, and were clothed in low-stature vegetation instead. In the middle of the Tertiary, however, Earth's climate became considerably drier and cooler. It is period known for radiation of mammals, and bird with flourishing of insects and angiosperms. Many lineages of flowering plants evolved herbaceous, non-woody forms. Grasslands spread over much of Earth. By the beginning of the Cenozoic era, invertebrate faunas resembled those of today. It is among the vertebrates that evolutionary changes during the Tertiary period were most rapid. Living groups of reptiles, including snakes and lizards, underwent extensive radiations during this period, as did birds and mammals. 3 waves of mammals dispersed from Asia to North America about 55 Maya. Rodents, marsupials, primates, and hoofed mammals appeared in North America for the first time. Some of the important evolutionary events in different epochs of Tertiary are as follows. Paleocene, major radiation of mammals, birds, and pollinating insects. Eocene, angiosperm dominance increases. Continued radiation of most present-day mammalian orders. Oligocene, origins of many primate groups. Miocene, continued radiation of mammals and angiosperms, earliest direct human ancestors. Pliocene, appearance of bipedal human ancestors. 5.1.2 The Quaternary, 1.8 Maya to present. The current geological period, the Quaternary, is subdivided into 2 epochs, the Pleistocene and the Holocene, also known as the Recent. The Pleistocene epoch was a time of drastic cooling and climatic fluctuations. During 4 major and about 20 minor episodes of glaciation, massive glaciers spread across the continents, and animal and plant populations shifted toward the equator. The last of these glaciers retreated from temperate latitudes less than 15,000 years ago. This retreat marked the beginning of the Holocene epoch. Organisms of the Holocene are still adjusting to these changes. Many high-latitude ecological communities have occupied their current locations for no more than a few thousand years. Interestingly, few species became extinct during these climatic fluctuations. However, the Pleistocene was the time of hominid evolution and radiation, resulting in the genus Homo and various species including Homo sapiens modern humans. Quaternary is known for megafauna extinction, as many large bird and mammal species became extinct in Australia and in the Americas when H. sapiens arrived on these continents about 40,000 and 15,000 years ago, respectively. Human hunting may have caused these extinctions, although existing evidence does not convince all paleontologists. 3.8 Key trends in history of life. 5.8.13 Major faunas have dominated life on Earth. The fossil record reveals 3 great radiations that resulted in the evolution of major new faunas. The first one, the Cambrian explosion, took place about 540 Maya. The second, about 60 million years later, resulted in the Paleozoic fauna. The Great Permian extinctions 300 million years later were followed by the third event, the Triassic explosion, which led to our modern fauna. During the Cambrian explosion, organisms representing all the major body plans of present-day lineages appeared, along with a number of lineages that subsequently became extinct. The Paleozoic and Triassic explosions resulted in many new groups of organisms, but all of them had modifications of body plans that were already present when these great biological diversifications began. 8.2 Variable rates of evolutionary change within lineages and fossils. In addition to revealing the broad patterns of the evolution of life on Earth, fossils also tell us about rates of change within particular lineages of organisms. The fossil record shows that no single pattern characterizes evolutionary rates. Many species have changed very little over many millions of years. Others have changed gradually over time. Still others have undergone rapid changes over short time periods, followed by long periods of slow change. Change may incorrectly appear to be rapid if the fossil record is very incomplete, but some rapid changes are well documented by excellent temporal series of fossils. Let's look at some examples of these patterns. 1. Some living species superficially remain unchanged during evolution. Species that have changed little over millions of years are known as living fossils. Fossilized leaves of the genus Ginkgo from the Triassic, for example, are very similar to those of living trees. Animals in some marine lineages have also evolved slowly. The horseshoe crabs living today are almost identical in appearance to those that lived 300 million years ago. The sandy coastlines where horseshoe crabs spawn feature extremes in temperature and salt concentration that are lethal to many organisms. These harsh environments have changed relatively little over millennia. The chambered nautiluses of the late Cretaceous are indistinguishable from living species. 2. Evolutionary changes have been gradual in some lineages. The fossil record contains many series of fossils that demonstrate gradual change in lineages of organisms over time. A good example is the series of fossils showing changes in the number of ribs on the exoskeleton in 8 lineages of trilobites during the Ordovician. Rates of change differed among the lineages, and they did not all change at the same time, but all of the changes were gradual. 3. Rates of evolutionary change are sometimes rapid. In the histories of some lineages, periods of gradual evolutionary change are broken by periods during which changes, either in the physical or biological environment, create conditions that favor rapid evolution of new traits. Such rapid evolutionary change is illustrated by the 3-spined stickleback, Gasterosteus aculeatus, a widespread marine fish that has repeatedly colonized new environments, freshwater lakes during its evolutionary history. Sticklebacks are tiny fish, usually less than 10 centimeters long. All marine and most freshwater populations have well-developed pectoral girdles with prominent spines that make it difficult for other fish to swallow them. However, large predatory insects can readily grasp the stickleback's spines, and they prey selectively on those stickleback individuals with the largest spines. When stickleback populations colonize freshwater habitats where predatory fish are absent, but predatory insects are present, they rapidly evolve smaller spines. Populations with reduced spines are found primarily in young lakes that were covered by ice during the most recent glaciation and do not have large predatory fish. The extensive fossil record of sticklebacks shows that spine reduction evolved many times in different populations that colonized fresh water. In addition, molecular data show that each freshwater population is most closely related to an adjacent marine population, not to other freshwater populations. At times, evolutionary change is rapid enough to be measured directly. A good example is provided by the house finch, a bird that as recently as 1939 was confined to the arid and semi-arid parts of western North America. That year, some captive finches were released in New York City. Many of them survived to form a small breeding population in the immediate vicinity of the city. During the early 1960s, that population began to grow and increase its range. By the 1990s, the house finch had spread across all of the eastern United States and southern Canada. Remarkably, by 2000, birds and finch populations that had been separated for only a few decades were as different in size as birds and finch populations that had been separated for thousands of years. 8.9. Mass extinctions. A species may become extinct because its habitat has been destroyed or because the environment has changed in a direction unfavorable to the species. If ocean temperature falls by even a few degrees, many species that are otherwise beautifully adapted will not survive. Even if physical factors in the environment are stable, biological factors may change. The environment in which a species lives includes the other organisms that live there, and evolutionary change in one species is likely to have some impact on other species in the community. 4. Example: the evolution by some Cambrian animals of hard body parts, such as jaws and shells, may have made some organisms lacking hard parts more vulnerable to predation and thereby more prone to extinction. Extinction is in fact inevitable in a changing world, and there have been crises in the history of life when global environmental changes have been so rapid and disruptive that a majority of species were swept away. In Figure 8 there is the series of peak times when extinction rates appear to be exceptionally high. These peaks are called as mass extinctions. From the Ordovician onwards, the 5 largest extinction events observed were in the late Ordovician, the late Devonian, the end of the Permian, the late Triassic, and the end of the Cretaceous. These are sometimes called the Big Five. Of these mass extinction chronicled in the fossil record, 2 have received the most attention. The Permian extinctions is the biggest in history, with 80 to 96% of species going extinct. The Permian extinction occurred about 250 million years ago, and the Cretaceous extinctions, in this extinction at least half, and perhaps 60 to 75% of species went extinct. The Cretaceous extinction occurred about 65 million years ago. Figure 39. Figure 39. Periods of mass extinction and the diversity of life. 5.9.1 Permian extinction. The Permian extinctions, which define the boundary between the Paleozoic and Mesozoic eras, claimed about 90% of the species of the marine animals. Terrestrial life also crashed. For example, 8 out of 27 orders of Permian insects did not survive into the Triassic, the next geological period. This mass extinction occurred in less than 5 million years, possibly much less in the context of geological time. Several factors may have combined to cause radical environmental change during the late Permian. That was about the time the continents merged to form Pangaea, which disturbed many marine and terrestrial habitats and altered climate. Massive volcanic eruptions also occurred in what is now called Siberia, constituting the most extreme episode of volcanism of the past half-billion years. Besides eruption of lava and ash into atmosphere, the Siberian volcanoes may have produced enough carbon dioxide to warm the climate. Reduced temperature differences between the equator and the poles may have slowed the mixing of ocean water, which in turn may have reduced the amounts of oxygen available to marine organisms. An oxygen deficit in the oceans may have played a large role in the Permian extinctions. 5.9.2 Extinction of dinosaurs. The Cretaceous extinction. Proposed in 1977 that Cretaceous extinction was caused by asteroid impact. Cretaceous tertiary border has high level of iridium, rare in Earth's crust but common in meteorites. Calculations of effects of nuclear bomb explosions, nuclear winter, compare with worldwide climate cooling expected from large asteroid impact. Worldwide layer of soot also defines iridium layer. Huge meteorite crater of correct age. Found in Caribbean Ocean and Yucatan Peninsula. Suspected site of impact of meteor that resulted in dinosaur extinction, marine animal. Fossil record indicates mass extinctions occur every 26 million years. Corresponds to movement of solar system within Milky Way galaxy. The Alvarez theory. Luis Alvarez, his son Walter, and their colleagues proposed that a giant asteroid or comet striking the Earth some 65 million years ago caused the massive die-off at the end of the Cretaceous. Presumably, the impact generated so much dust and gases that skies were darkened all over the Earth, photosynthesis declined, and worldwide temperatures dropped. The outcome was that as many as 75% of all species, including all dinosaurs, became extinct. The key piece of evidence for the Alvarez hypothesis was the finding of thin deposits of clay containing the element iridium at the interface between the rocks of the Cretaceous and those of the Tertiary period, called the KT boundary. Iridium is a rare element on Earth, although often discharged from volcanoes, but occurs in certain meteorites at concentrations thousands of times greater than in the Earth's crust. After languishing for many years, the Alvarez theory gained strong support from the discovery in the 90s of the remains of a huge, 180 kilometers in diameter, crater in the Yucatan Peninsula that dated to 65 million years ago. The abundance of sulfate-containing rock in the region suggests that the impact generated enormous amounts of sulfur dioxide, SO2, which later returned to Earth as a bath of acid rain. A smaller crater and low is formed at the same time. Many have contributed to the devastation. Perhaps during this period the Earth passed through a swarm of asteroids or a comet, and the repeated impacts made the Earth uninhabitable for so many creatures of the Mesozoic. 5.9.3 Extinction due human activities. There is little doubt left in the minds of professional biologists that Earth is currently faced with a mounting loss of species that threatens to rival the 5 great mass extinctions of the geological past. Harvard biologist E.O. Wilson estimated that Earth is currently losing something on the order of 30,000 species per year, which breaks down to the even more discouraging statistic of some 3 species per hour. Some biologists have begun to feel that this biodiversity crisis this 6th extinction is even more severe and more imminent than Wilson had supposed. Can we stop the devastation of our planet and save our own species? We are in a biodiversity crisis, the fastest mass extinction in Earth's history, largely due to 1. Human destruction of ecosystems, 2. Overexploitation of species and natural resources, 3. Human overpopulation, 4. The spread of agriculture, and 6. Pollution. At first glance, the physically caused extinction events of the past might seem to have little or nothing to tell us about the current 6th extinction, which is a patently human-caused event. And because Homo sapiens is clearly a species of animal, the 6th extinction would seem to be the first recorded global extinction event that has a biotic, rather than a physical, cause. 5.9.4 Extinction rates vary over time. More than 99% of the species that have ever lived are extinct. Species have become extinct throughout the history of life, but extinction rates have fluctuated dramatically. Some groups had high extinction rates while others were proliferating. A mass extinction changes the biota of the following period by selectively eliminating some types of organisms, thereby allowing others to increase in their relative abundance. For example, among the mollusks of the Atlantic coastal plain of North America, species with broad geographic ranges were less likely to become extinct during normal periods, when no mass extinctions were taking place, than were species with small geographic ranges. During the mass extinction of the late Cretaceous, however, groups of closely related mollusk species with large geographic ranges survived better than groups with small ranges, even if the individual species within the group had small ranges. Similar patterns are found in other mollusks elsewhere, suggesting that traits favoring long-term survival during normal times are often different from those that favor survival during times of mass extinctions. At the end of the Cretaceous period, extinction rates on land were much higher among large vertebrates than among small ones. The same was true during the Pleistocene, when extinction rates were high only among large mammals and large birds. During some mass extinctions, marine organisms were heavily hit, but terrestrial organisms survived well. Other mass extinctions affected organisms living in both environments. These differences are not surprising, given that major changes on land and in the oceans did not always coincide. 5.10 Evolutionary developmental changes. The depth of changes that occurred in the history of life can be determined from fossil records. Understanding about plate tectonics, mass extinction, and adaptive radiation reveal more about how those changes occurred. Although the main cause of changes is environmental or geologic alterations, they occur due to genetic changes that are permanent. These genetic changes are the intrinsic biological mechanism of evolution of life. For this, we turn to genetic mechanisms of change, paying particular attention to genes that influence development. 5.10.1 Effects of developmental genes. Slight genetic changes can give rise to major morphological differences. This can be estimated by research at the interface between evolutionary biology and developmental biology. There are certain genes which have a major role in development of an organism, as they control rate, timing, and spatial pattern of change in an organism's form as it develops from a zygote into an adult. Alterations in such development-related genes lead to major changes in the life forms. 5.10.2 Changes in rate and timing. An evolutionary change in the rate or timing of developmental events is called as heterochrony, due to which many striking evolutionary transformations occur. For instance, the shape of an organism depends on the relative growth rates of different body parts during development. Thus, changes in the growth rates alter the adult forms dramatically, as seen in the contrasting shapes of human and chimpanzee skulls. Figure 40. Figure 40. In the human evolutionary lineage, mutations slow the growth of the jaw relative to other parts of the skull. As a result, in humans, the skull of an adult is more similar to the skull of an infant than is the case for chimpanzees. Some other examples of the dramatic evolutionary effects of heterochrony include increased growth rates of finger bones in bats, which results into the skeletal structure of wings, and reduced growth of leg and pelvic bones in whales, which eventually led to the reduction and subsequent loss of hind limbs in whales. The timing of reproductive development relative to the development of non-reproductive organs is also regulated by heterochrony. If the organs involved in reproduction develop faster than the non-reproductive organs, then the sexually mature stage of a species may retain body features that were juvenile structures in an ancestral species, a condition called pedomorphosis. From the Greek paidos, of a child, and morphosis, formation. For example, most salamander species have aquatic larvae that undergo metamorphosis in becoming adults. In case of certain other species, non-reproductive organs grow faster to attain the adult stage, and then become sexually mature while retaining gills and other larval features. In such cases, even a small number of genetic changes during development produce organisms that appear very different from their ancestors. Indeed, recent evidence indicates that a change at a single locus was probably sufficient to bring about pedomorphosis in the axolotl salamander, although other genes may have contributed as well. 5.10.3 Changes in spatial pattern. Changes in the genes that control the placement and spatial organization of body parts can also result in substantial evolutionary changes. For example, homeotic genes are master regulatory genes which determine some basic features such as location of particular organ, i.e., where a pair of wings and a pair of legs will develop on a bird's body plan, or how a plant's flower parts are arranged, etc. The homeotic genes are thus called as organ identity genes. The positional information in an animal embryo is given by products of one class of homeotic genes, called as the Hox genes. Due to the Hox gene, cells become able to develop into structures appropriate for a particular location. Alteration in the Hox genes or their mode of expression can have a profound impact on morphology. For example, among crustaceans, a change in the location where two Hox genes, UX and SCR, are expressed correlates with the conversion of a swimming appendage to a feeding appendage. Figure 41. Figure 41. Origin of insect body plan. Large effects are also seen in snakes, where changes in how two Hox genes, HOXC6 and HOXC8, are expressed suppresses limb formation. Similarly, when comparing plant species, changes to the expression of homeotic genes known as MADS box genes can produce flowers that differ dramatically in form. 5.10.4 Evolution is continuous, but not goal-oriented process. What we understand from macroevolution studies is that the origin of new species throughout the history of life has been affected by various factors. These factors include both the bottom-up factors, such as natural selection operating in populations, and the top-down factors described here, such as continental drift promoting bursts of speciation throughout the globe. According to Francois Jacob, evolution is a process in which new life forms arise by the slight modification of existing forms. Even large changes, like the ones that produce the first mammals or the six-legged body plan of insects, can result from the modification of existing structures or existing developmental genes. Over time, such tinkering has led to three key features of the natural world: the striking ways in which organisms are suited for life in their environments, the many shared characteristics of life, and the rich diversity of life. 5.10.5 Evolutionary Innovations. Francois Jacob's view of evolution reminds the Darwin's concept of evolution, i.e., descendants with modifications. According to the concept, gradual modifications in the ancestral structures give rise to novel and complex structures to form new species. In many cases, the newly formed complex advanced structures, which are evolved from simpler versions, also perform the same basic functions. In case of human eye, it is an intricate organ which is made from numerous parts that work together in forming an image and transmitting it to the brain. If human eye have been evolved in gradual increments, and if it needs all of its components to function, a partial eye could not have been of use to our ancestors. Darwin himself noted this flaw in his theory, as it lies in the assumption that only complicated eyes are useful. In fact, many animals depend on eyes that are far less complex than our own. Figure 42. The simplest eyes are made up of patches of light-sensitive photoreceptor cells. These simpler eyes are found in a variety of animals, including small mollusks called limpets, and appear to have a single evolutionary origin. These eyes cannot focus the images they visualize, but can distinguish objects in light from those in dark. Limpets have developed a behavioral adaptation which reduces their chances of being eaten. For this, they cling their rock more tightly when a shadow falls on them. Because limpets have had a long evolutionary history, we can conclude that their simple eyes are quite adequate to support their survival and reproduction. Complex eyes of the animal kingdom have been evolved independently form some basic structures. In case of some mollusks, such as squids and octopuses, the eyes are as complex as the human eyes and those of other vertebrates. Figure 42. Although complex mollusk eyes evolved independently of vertebrate eyes, both evolved from a simple cluster of photoreceptor cells present in a common ancestor. If the serial, incremental modifications in each case benefited organism, the eyes persisted with the organism. Figure 42. A range of eye complexity among mollusks. The independent evolution may also be evident in their structures. Vertebrate eyes detect light at the back layer of the retina and conduct nerve impulses towards the front, whereas the complex mollusk eyes do exactly opposite of this. They detect the light at front layer of retina and conduct the nerve impulse towards backside. Though the mode of action is different, throughout their evolutionary history the basic function of vision is retained. Sometimes evolutionary novelties can also arise when structures that originally played one role gradually acquire a different one. For example, as cynodonts gave rise to early mammals, bones that formerly comprised the jaw hinge, the articular, quadrate, and hyomandibular were incorporated into the ear region as ear ossels of mammals, where they eventually took on a new function, the transmission of sound. Ancestral jaw bone and derived ear ossicle are as follows. Structures, which initially perform one function but eventually become co-opted for another function, are sometimes called adaptations to distinguish them from the adaptive origin of the original structure. The term adaptation does not imply that a structure somehow evolved in anticipation of future use, because natural selection can only improve a structure in the context of its current utility, and it cannot predict the future. Novel features can arise gradually and not purposefully. For an instance, the new jaw hinge and ear bones of early mammals were not purposefully designed, but they were gradually formed in a series of intermediate stages, each of which has some function in the organism's current context. 5.11 Evolution of horse life has been observed to follow certain patterns or trends throughout evolution. For instance, in case of some evolutionary lineages, increased or decreased body sizes are favored. 55-million-year-old Hyracotherium is the ancestor of present-day horse, genus Equus, figure 43. For example, the horses are one of the oldest forms of mammals. Evolution of horse dates back to Eocene epoch, more million than 50 million years ago. Primary center of evolution were Great Plains of North America, from where species migrated to Europe and Asia from time to time. Horses became extinct in North America by the end of Pleistocene epoch, but their offshoots in Europe and Asia flourished. Evolution of horse was triggered by a change in the climate and vegetation during Lower Cenozoic period, when grasslands in most parts of the world replaced forests. The main modifications in the body of horses can be outlined as follows. 1. Increase in the size and height of the body from a small, rabbit-like animal to 6 feet tall grassland animal. 2. Gradual enlargement and better development of the third digit, median digit, and reduction of the other lateral digits. 3. Lengthening of the limbs and perfection of the hoof for fast running in open grasslands. 4. Reduction of ulna bone in the foreleg and fibula in the hind leg and strengthening of radius and tibia. 5. Change from digitigrade to unguligrade, locomotion for fast running. 6. Elongation of the preorbital or facial region of the skull and migration of eyes to the top of head. 7. Modification of teeth from brachydont, low-crowned, to hypsodont, high-crowned, to withstand tougher food, grass. 8. Increase in the size and complexity of the brain for superior intelligence. 9. Reduction in pectoral girdle and disappearance of the weak clavicle. 10. Body became streamlined, muscles tight, without loose V-fat, for long and sustained running. 11. Nostrils became wide to allow more air into strong lungs and stamina increased. According to the fossil records, the evolution of modern horse cannot be represented in a single evolutionary progression. However, it is like describing a bush as growing toward a single point by tracing only the branches that lead to that twig. For example, by selecting certain species from the available fossils, it is possible to arrange a succession of animals intermediate between Hyracotherium and living horses that show a trend toward large, single-toed species. However, the genus Equus did not evolve in a straight line. It is the only surviving twig of an evolutionary tree that is so branched, more like a bush. A series of various speciation episodes included several adaptive radiations before emergence of Equus. Not all of them were directing towards a large, one-toed, grazing horse. Figure 43. The evolution of horses. Using light color to trace a sequence of fossil horses that are intermediate the present-day horse, Equus, and its Eocene ancestor Hyracotherium creates the illusion of a propensive sign, reduced number of toes, and teeth modified for grazing. The important stages in the evolution of horse is summarized below. Backhand index pointing down. 1. Eohippus, genus Hyracotherium. Hyracotherium had a size of a large dog/deer, had 4 toes on its front feet, 3 toes on its hind feet, and teeth adapted for browsing on bushes and trees. It is nicknamed as dawn horse. It lived about 56 million to 33.9 million years ago. Its toes ended in a strong, thick, horny type nail in their tips. Eohippus had little or no lateral vision, and teeth were similar to a pig, short and crowned for eating plants. Its skin color allowed to camouflage. 2. Mesohippus. It has longer snout, legs, and neck compared to Eohippus. It lived around 35 to 40 million years ago and had less arched back. It has 3 toes on each foot. Larger limbs and legs caused the Mesohippus to run faster, eliminating the need for camouflage. 3. Myohippus, ankle joint changed, stood at a minimum of 24 inches at the shoulder, weighed much more than the Mesohippus, head longer, and incisor teeth formed. Mesohippus and the Myohippus species overlapped for more than 4 million years until another change occurred. For Merychippus, it exists 30 million years ago, looked very similar to today's horses, and stood over 36 inches tall at shoulder. It still had 3 toes, outside toe became weaker, head changed, the eye moved, allowing better vision. It has longer neck, for easier grazing, developed defenses and better sense of smell. In it, toes started turning into hooves, and teeth developed into the teeth we know today. 5. Pilohippus. It existed around 12 to 6 million years ago and considered as direct link to the equus, which is, in short, the horse we know today. It is first single-toed, or hoofed horse, had strong leg ligaments to increase speed and power. It had dished face and resembled a ponyline. Many features. 6. Equus. It is modern horse which appeared first time around 5 million years ago. It does not have a dished face and have only 1 toe on each foot and possess teeth modified for grazing on grasses. Many strands of horses arose after the Pilohippus. The equus was the only to survive. 5.12 Evolution of human. Evidences suggest that most of hominid evolution occurred in Africa and Asia, whereas the entire evolution of the human species took place in Africa. The important transitions in hominid lineage are represented in Table 6. Fossil records suggest that there were several species belonging to the genus Homo. For example, Homo habilis lived in Africa about 2 million years ago and was characterized by having a larger brain than Australopithecus, using tools and being bipedal. Homo erectus was another species which used fire and migrated to Asia and Europe, seems to be appeared about 1.7 million years ago. The fossils of Homo erectus are popularly called as Java man and Peking man. Later on, Homo sapiens replaced Homo erectus. Figure 44. Figure 44. The evolutionary relationship among hominid species. A primitive form of Homo sapiens, called Neanderthal man, Homo sapiens neanderthalensis, and Denisovan, Homo sapiens denisova, were common in Europe and Asia, and it is believed to be originated from Homo heidelbergensis. Figure 45. Despite of having relatively short and stocky and more powerfully built up, the Neanderthal man resembled modern man the most. The Neanderthals used to build hut-like structures for dwelling, made tools, and used animal hides as clothing, and also used to bury the dead individuals. An abrupt transition occurred all over Europe, evidence that the Neanderthal man was wiped out, and its close, more efficient cousin, the Cro-Magnon, evolved about 34,000 years ago. Table 6. The summary of human evolutionary lineage. These Cro-Magnon people were more advanced than any of the previous transitions. They developed a cultured lifestyle and left behind very elaborate cave paintings dictating about the same. Modern man Homo sapiens took charge after the last glacial period. About 10,000 years ago, they spread all over the globe, cultivated plants, domesticated animals, and reached enormous population sizes. Evidences reveal that Homo sapiens appeared in Africa about 500,000 years and might have replaced Homo erectus there. But in Asia, Homo erectus appears to have survived for another 250,000 years when it was finally replaced by Homo sapiens migrating from Africa. Recently, fossils of a small-bodied hominid species Homo luzonensis was discovered. It ventured into the islands of Southeast Asia from mainland Asia about 67,000 years ago. This species is believed to be closely related to another Homo species discovered previously from an island in Indonesia. Out of Africa hypothesis and the multi-regional hypothesis. Broadly speaking, there are two competing hypotheses on the origin of modern humans: the out of Africa hypothesis and the multi-regional hypothesis. Both agree that Homo erectus originated in Africa and expanded to Eurasia about 1 million years ago, but they differ in explaining the origin of modern humans, Homo sapiens sapiens. The multi-regional hypothesis states that independent multiple origins with continuous gene flow between continental populations, figure 46a, occurred in the million years since Homo erectus came out of Africa. According to Tini's hypothesis, Homo sapiens evolved in each of the regions where its fossils are now found from ancestral Homo erectus that migrated out of Africa about 1.5 million years. Advocates of this hypothesis consider H. erectus to be an early version of H. sapiens and not a different species. Constant interbreeding between neighboring populations of this archaic Homo sapiens may have prevented reproductive isolation, resulting in our present-day races of Homo sapiens, rather than multiple species of Homo. The second hypothesis, out of Africa, proposes that a second migration out of Africa happened about 100,000 years ago, in which anatomically modern humans, Homo sapiens, of African origin conquered the world by completely replacing descendants of the earlier H. erectus migrants. Figure 46b. Mitochondrial DNA, mtDNA, and Y chromosome DNA evidence support the out of Africa hypothesis. A compromised version of the out of Africa hypothesis emphasizes the African origin of most human populations, but allows for the possibility of minor local contributions. Important key points: Azoic arid era of invisible life. Archaeozoic arid era of former life or era of early life. Paleozoic arid era of old life or era of ancient life. Mesozoic era of middle life, also called Age of Reptiles. Cenozoic era of recent life, also called Age of Mammals, Birds, Insects, and Angiosperms. Ordovician period. Age of Invertebrates. And origin of amphibians. Devonian period. Age of fishes and of origin of amphibians. Carboniferous periods. Age of amphibians. And origin of reptiles. Earliest reptiles evolved in Carboniferous period. Jurassic period. Age of reptiles. Holocene epoch. Age of man. First vertebrates, Ordovician. First amphibian, Devonian. First reptiles, Carboniferous. First mammals, Triassic. First birds, Jurassic. First humans, Quaternary, Pleistocene. First human, Quaternary, Pleistocene. First amphibian plants, Bryophytes, Ordovician. First land plants, Pteridophytes, Silurian. First seed plants, Gymnosperms, Devonian. First flowering plants, Angiosperms, Cretaceous. Rules to remember. Cope's law. It states that warm-blooded animals, population lineage, tend to increase in body size during the long course of evolutionary time. Dollo's law of irreversibility, also known as Dallow's law and Dallow's principle. It states that an organism never returns exactly to a former state, even if it finds itself placed in conditions of existence identical to those in which it has previously lived. Organisms always keep some trace of the intermediate stages through which it has passed. Occam's principle. It states that suppose two explanations for an occurrence. In this case, the simpler one is usually better. Another way of saying it is that the more assumptions you have to make, the more unlikely an explanation. Van Valen's law. Van Valen proposed the Red Queen hypothesis, 1973, as an explanatory tangent to the law of extinction. The Red Queen hypothesis captures the idea that there is a constant arms race between co-evolving species. Amongst other work, Van Valen's proposed law of extinction, known as Van Valen's law, drew upon the apparent constant probability, as opposed to rate, of extinction in families of related organisms, based on data compiled from existing literature on the duration of tens of thousands of genera throughout the fossil record. Williston's rule. During evolution of lineage, serially homologue parts tend to reduce in number, but get more and more differentiated, e.g. prawn's leg.